This is the video number 3 for the mouse genetic approaches for neuroscience lecture. In this lecture, we will talk about targeted gene expression, constitutive or inducible conditional animals, in which we will specifically target the mutation to a body region or a cell type specific. For site-specific recombination, we use the Cre LoxP system, and we use conditional gene targeting by using the Cre LoxP system from the bacteriophage P1, which is probably the most widely used site-specific recombination tool. This system uses a recombinase enzyme called Cre, which mediates recombination between two specific sequences called LoxP sites. These LoxP sites are small bacterial sequences around 34 base pairs. There are inverted repeats that confer orientation, and they can be used by the Cre enzyme to remove anything that is contained between these two LoxP sites. The recombinase Cre takes these two LoxP sites and excises or removes anything that is contained between those sites. This is an example of a gene construct for conditional targeting, in which we will flank in particular exon of a target gene with LoxP sites to enable conditional targeting when combined with the specific Cre system that we will see in the next slide. In this construct, what we use is we will use the 5' homologous arm that contains the exon 1 of the gene. We will also generate an exon 2 that is flanked with these LoxP sites, followed by the neocassette to select for positive targeting. And this will be followed by a 3' homologous arm that will enable recombination with this other side of the genomic DNA. After homologous recombination, we will remove exon 2 and replace it by a LoxP flanking sites of the exon 2. Under normal conditions, this gene X will be transcribed normally, but when combined with a specific Cre system, the Cre system will excise this exon 2. In this case, this is a gene targeting using the Cre LoxP system. As we said before, we will have to generate a conditional animal in which a particular gene or a fraction of this gene is flanked by the LoxP sites. Then we will have to crossbreed this animal with a mouse that contains the recombinase Cre that is expressed under a cell-specific promoter. As a result of this, we will have an animal that contains both the gene flanked by the LoxP sites and the recombinase Cre expressed in a particular cell type that is controlled by a specific promoter. Then recombination takes place, and this animal will have gene A deleted in a specific cell type where Cre is expressed. This is another example of using the Cre LoxP system, but for generating inducible gene targeting. For this, we will need a modified version of the Cre enzyme, which is linked to the estrogen receptor. Estrogen receptor can be activated by the use of tamoxifen, and the use of tamoxifen will drive the localization of Cre into the nucleus. So this system allows for external temporal control of the Cre activation. For this example, we will generate a mutant animal in which we inserted this construct. We use a bacterial constitutive plasmid promoter that contains a stop codon that is flanked with LoxP sites, followed by a GFP protein that is fused to an additional gene that we want to express under certain circumstances. We will also combine this animal with a Cre that is controlled under a cell-specific promoter. In the absence of tamoxifen, Cre is expressed but is retained in the cytoplasm because it cannot get into the nucleus. Therefore, this product will not be expressed. In the presence of tamoxifen, now the drug binds to the estrogen receptor part of the Cre and drive the enzyme into the nucleus. Now the Cre enzyme can remove the LoxP sites that flank this stop codon, and now the R26 promoter will drive the expression of this transgene chimera, which is a GFP protein fused to an additional gene. So by this system, we can control the expression of this additional gene fused to GFP whenever we want and wherever we want in the presence of tamoxifen. Here there is a problem that we will see in class, in which you have to design an experiment to generate a knockout mouse lacking irregular NRG1 specifically in astrocytes. For that, you can use the promoter of genes that are markers for astrocytes. And here there are different examples of promoters for genes that are exclusively expressed in astrocytes. You could also consider adding a reporter to control for expression specificity. And in this problem, we will see different concepts that we've learned in previous videos and new other concepts that we've seen in this video. And I really encourage you to think about this in advance, and then we will do this problem in class.